Hello and welcome to the webinar where we will present a challenging typing case and show you the power of our NGS Engine software. Before we start, I will briefly introduce myself and the speaker of today. My name is Veerle van Meerkant and I am Technical Support Specialist at Gendex. As a member of the support team, I help our customers with their questions, and together with the support team, we also organize all Gendex educational events. Through our monthly webinars, we want to share with you our expertise. The presentation will be about 25 minutes and we take live questions during and after the webinar. If you have any questions, please type in your question in the chat box of the GoToMeeting application. Our webinars are part of the Gendex Training School, which is an APHI accredited provider of continued education. Besides the webinars, we organize several other educational events throughout the year, such as the one-day teaching sessions before the start of the two large immunogenetics conferences, EFI in Europe and ESHI in the US in September. And we also offer 3 days hands-on training courses which provide you with the confidence to start up HLA typing or chimerism monitoring in your own laboratory. These are customized training opportunities, so whether you're a beginner or advanced, we tailor to your needs. There are several opportunities throughout the year in both Europe and the USA. And now I would like to introduce the speaker of today, my colleague Hanneke Merkes, who is Associate Director Support and Education. Hanneke joined Gendex in 2015. As a support team director, she has seen many interesting typing cases passing by and she is a true HLA expert. For this webinar, she has selected a very interesting typing case that was shared with the support team by a customer. Hanneke?
Thank you very much, Veerle. So I would like today to present our webinar, The Power of NG Engine, Unravelling 3 Alleles in a Clinical Sample. At Gendex, we have a support team every day available to answer every question that comes from customers. This can happen either by phone or by sending an email, and we thrive to answer these questions within 1 working day. Because we value questions and feedback of customers, we encourage everyone to share data with us when they have a question. This can be done by the following link on the website, and you can share your data with support. We are happy to see that many customers reach out for us for feedback and advice. During this webinar, I would like to discuss a very challenging case that reached support. The customer uses Gendex SBT Accelerator and to type HLA of clinical samples of DNA derived from blood using Sanger sequencing. They share their data with Gendex support for advice. This procedure first requires an amplification of each locus. This is followed by an heterozygous sequencing reaction, sequencing exon 2, 3 and 4, both forward and reversed, and ambiguities that remain are resolved by a second round of sequencing, but now with a GSSP, a group-specific sequencing primer, which only sequences one of the 2 alleles and results in a homozygous sequence result. We represent here the data of the forward and the reversed sequencing reaction of exon 3, analyzed in SBT Engine. As you can see from the screenshot, was the data nice and clean, and the heterozygous positions are represented by the double peaks. To come to an unambiguous typing, a second round of sequencing was required to assign the heterozygous positions to either one of the 2 alleles. Therefore, a second round of sequencing was performed, and in this case with 2 genotype-specific sequencing primers, or GSSP, where each GSSP is specific for one of the 2 alleles. Analysis of the first GSSP, the A25, as shown on top, clearly sequenced allele 1 and resulted in a clean homozygous sequence result. The result of GSSP A23, however, was unexpected. Although the overall majority of the sequence was homozygous, there was 1 position that remained heterozygous. The sequence results clearly indicate a double peak from a C and a T, although only a C was expected. This was only observed for this 1 position in exon 3. All the other positions were clearly homozygous. We could not explain these results and therefore offered to sequence the sample with NGS to investigate what could have happened. From signer sequencing so far, we now know that the GSSP specific for allele number 1 showed a clean homozygous result for exon 3, and the other GSSP specific for allele 2 showed a heterozygous position with a C and a T. We therefore sequenced this sample with next generation sequencing. We first made a library with NGS-Go and subsequently sequenced it on the Illumina MySeq, and the data was analyzed in NGS Engine. When we analyze with NGS Engine, we obtain the following parameters in the overview screen. The sample has a high locus mappability, has excellent read depth, and the best match is a common typing. And there are no intron and no exon mismatches with this typing in the IMGT database. At last, there was only one phasing region. The only parameter here is indicated in red which drew our attention. NGS Engine indicated here that there's one position in a core region of HLA-A that is unclear, and it was visualized by a red question mark. To get an indication of the quality of the data, we switch on our quality metrics. You can do this in NGS Engine via the box indicated with the arrow, and we choose to inspect the quality in the core plus and the exon plus. When the quality metrics are shown in NGS Engine, all values get a color as an indication of the quality. When data is of low quality, this will be flagged red. Data with intermediate quality will get a yellow color, and all high quality data will remain uncolored. We also observe again one position in red with a question mark, indicating that one position in the core region is unclear. So we conclude that the HLA locus is sequenced with high quality data in the exon regions and that one position remains unclear. We therefore inspect the HLA locus into more detail by clicking on the blue HLA-A. This brings us to the overview screen of the HLA locus. On the top is the HLA gene represented, with the UTRs in blue, the exons in yellow and the introns with the black lines. Just below the gene is the overall coverage and read depth indicated. The vertical lines represent the SNPs or the heterozygous positions. In this example, they are nicely distributed across the gene. In the beginning of the third yellow box, representing exon 3, we see again a red question mark. And to zoom in on this position, we can click on the blue arrow below that directly brings us to mismatches or question marks. We now have zoomed in the beginning of exon 3. I will first talk you through what you see in the screen. On the top, we have our sequence results, and all the sequence spaces are indicated and the heterozygous positions as well. Just below our results, you can observe the IMGT database with the sequences of the best matching results. And below that are gray lines that represent the individual reads for the alleles. The top panel is reserved for the reads from allele 1, and the bottom panel is reserved for the reads from allele 2. These base calls are gray because the data matches with the IMGT reference. When a sequence result does not match the IMGT, they will be represented by a letter. NGS Engine gives you a tool to inspect every positioning manually if you would like. We can do this by example for the heterozygous position over here. When we mouse over this position, there's a screen that pops up. And in this screen, you get information on this position. And this also includes the information how often the position is sequenced and which bases you sequenced exactly. And in this example, we see that we sequenced 333 times an A and 289 times a G. This makes it a very nice heterozygous position in a balance of 53 to 46%. Now we will use the same tool to inspect the unclear position, indicated with the question mark. When we mouse over this position, we see that it sequenced both a C and a T, but not in a nice balance as we just observed before. The C is represented in 31% of the results, while the T is represented in almost 70% of the results. And when we scroll through the individual reads, we see that for allele 1, all reads contain a T, and for allele 2, we find both T's and C's. To get a further understanding of what is happening in this sample, we inspected the base variation plot. This is a plot that is the most important graph in NGS Engine, and it gives you a very clear picture of the quality of your data. Please notice that we use the same yellow color for color coding the exons. This graph showed the percentage of the most frequent base call. The most frequent base call is marked in blue. This means that for every position that is a true homozygous position, the most frequent base call will be at 100%. You will find such a position with the blue X on the 100% line. When a position is a perfect heterozygous, the most frequent base call is on the 50% line, indicated by an X in blue. The second base call is colored red and also indicated by a capital X on the 50% line. Please do note the logarithmic scale. All PCR imperfections and sequence noise will be presented by little x's on the bottom, around the 1 to 5%. This is your noise. When we look at the base variation plot of this particular sample, we can see that the heterozygous positions are all around the 50%. The blue axes represent the most prominent allele, allele number 1, and are approximately 53% of the reads. The second allele is just below that one on approximately 47%. But there is one position at the beginning of exon 3 that does not follow these lines at all. This position is represented by an orange dot, indicating that it's a question position, and it's represented in 30% of the reads. This confirms our previous observations that a subset of the reads has a variation in the sequence at the beginning of exon 3. So to summarize our observations so far, we've sequenced a sample for HLA-A, and this resulted in data of excellent quality. It resulted in a heterozygous sample where the two alleles are represented in a ratio of approximately 47 to 53%. There is, however, one position in exon 3 that does not follow this ratio. When we inspect this specific position for both alleles, we see that for allele 1, we found back that position has a T, but for allele 2, we see both a T and a C. And since we can see that for allele 2 there are two base calls, we wanted to do the two following steps. We want to confirm that we're not looking at noise, and we want to quantify the percentages of both alleles. To be able to investigate this effect of this variation, we can edit the position to heterozygous. You can do this by right mouse clicking on the data and subsequently selecting heterozygous. When you then move back to the overview screen, you can see that the button has changed to Apply Edits. When you click on Apply Edits, the sample will be reanalyzed and now considering the unclear position as heterozygous. When we applied the edits, we observed three changes. First, a little pencil appeared, which indicates that we made an edit. The best matching genotype is indicated in red because there's now a mismatch with an exon region, and the question mark has disappeared. When we then look into more details of the HLA locus, we again see a little pencil on the position indicated that the position is edited. There's also a small triangle appearing with the IMGT library. This is indicating that the data from the IMGT for the 0201 does not match with our data on this position. To explain the phasing, I would first like to show you an example of excellent phasing. I would like to therefore focus on the two heterozygous positions in this box. When we right mouse click on the location of the star in between the box, we can see a pop-up screen. This screen gives us all the information we need on the phasing between these two heterozygous positions. On the left, there's an AG homozygous, and on the right, there's a TG homozygous. And there are in total 548 reads that cover these two positions. From all the reads, there are almost 300 reads that are having an A in combination with a T. and there are almost no reads where the A is in combination with the G. We therefore know for sure that the A and the T are together in the same allele. The same works for the other base. With the G on the left, this G is together with another G in almost 250 reads. This means that we're absolutely sure that the two G's belong together in the same allele. This is an example of perfect phasing, and we can clearly assign the two heterozygous positions to the two alleles. When we do this for our unclear position in exon 3, together with the heterozygous position on the left, we see the following table. On the left there's an A and a G, and on the right there's the unclear position. There are 541 reads that cover both positions, and when there's an A on the position on the left, we see that there are 293 reads with a T and no reads at all with the C. That means that all A's of the 0101 belong together with a T, no exceptions. When there's a G on this position, there are two options. 80 reads have a T and 167 reads have a C. And we can quantify this to percentages of 15% and 31% of the total reads. So now we can add to our previous conclusion for position 373 that the A allele 1 has a T in 54% of the cases and allele 2 has two variations. The O201 with the T is 15% of the reads, but the variation with the C is in 31% of the reads. And because this position is fully phased, we can rule out that this is noise. Because if it would have been noise, the C would have been randomly distributed across both alleles. In the last step, we wanted to investigate what was the consequence of this mutation. In NGS Engine, you can visualize the amino acids from the HLA by clicking on the codons over here. You will see that on the top the codon will appear and indicating the appropriate amino acid. When we look at our position 373 in exon 3, we see that it concerns the first base of a codon. In the case of a T on this position, the codon will be TGC, which codes for a cysteine. When there is a C on this position, it will code for CGC, which will code for an arginine. So this means for allele 1, the codon will be coding for cysteine, but for allele 2, it will be either a cysteine or an arginine. So what are the steps that we've taken during this study? We've looked at Sanger data for this sample, and this showed us that allele 2 has a variation. With NGS Engine, we have expected the individual reads, and this revealed 3 alleles. Base variation plots confirmed these 3 alleles, and by investigating the phasing, we ruled out noise and we could quantify. And we investigated the consequences of the mutation. So, in conclusion, this is a very nice example that demonstrates the advantages of NGS. We hope to have convinced you now that the sample that contains multiple alleles for HLA, with the first being an O1 and 101, and the second allele consisting of 2 alleles, an O2, O101, and a new variant of an O2. And this new variation results in a coding variation of a cysteine to an arginine. We also looked asked for the clinical background of this interesting sample, and the lab was so kind to share with us that this sample was taken from a patient diagnosed with an AML. The lab also performed the typing on a sample that did not originate from the blood, and in this case they used a buccal swab. This buccal swab did not show the new variant, and therefore we hypothesized that the DNA of this sample contained both healthy and tumor cells, and that the tumor introduced a mutation into the HLA gene.
Thank you, Hanneke, for sharing this interesting typing case, and thank you for listening to our webinar. We hope to see you next time.